Thank you. My name is Miguel Fernandez, and I'm here to present what has been the start of my PhD at the Barcelona Supercomputing Center. As I have been introduced, we titled this Dynamic and Energetic Consequences of Disulfide Bonds in Proteins. So it's only natural that I start by explaining what a disulfide bond is, which is basically a covalent bond that is formed between two cysteines when they are oxidized. And the consequence in the protein structure is basically that we bring together different parts of the structure, and typically this three-dimensional conformation changes. So since we're talking protein structures, I think it's a good moment to say that proteins, as have been said today, have evolved to be optimized, not only to fold and be very energetically stable, but also to function. And this is more or less the principle of minimal frustration that has been explained a little bit today. But I want to explain you how we can measure actually this frustration. So you take your native energy, your native state of a protein, and you measure the energy associated to either a contact or a residue. Then you make a lot of random decoys, and then you measure the energy of these decoys and you build a distribution of energies. Now, frustration is just as easy as a comparison between your original energy and this distribution. So take your starting contact or residue, and it can be either less stable than most of the decoys, and this would be highly frustrated. It can be more stable than most of the decoys, and this would be minimally frustrated, or it can be just as stable as one would assume by looking at the distribution. And we would say that this is neutral frustration. And now that we clear this, I think it's pretty easy to understand that for foldable proteins that are generally minimally frustrated, the energetic landscape is funneled to and has a very strong bias towards the native state. But there are other situations in which this landscape is much more frustrated and therefore flat, and there are many structures with similar energies competing for the native state. So the native state is actually a conformational ensemble, and this is what we call an intrinsically disordered protein, because it doesn't have a three-dimensional structure that is stable. I explain all of this because basically we've already used frustration to understand many things about proteins, like binding sites, allosteric sites, or active sites and enzymes. And we suggest to do the very same with disulfide bonds. So basically, what we want to do is to answer this question. Can we better characterize the interplay between disulfide bonds, disorder, and frustration? So there's only really one question left to answer, and it is, what do disulfide bonds do in this context of frustration and disorder? So we did a little bit of background check on them, and we found out that there are three possible scenarios for it. We can see disulfide bonds helping the folding of very small proteins that in other situations wouldn't be able to fold, and this has some energetic consequences in the structure. In other situations, we've seen disulfide bonds take these proteins that do not have a stable three-dimensional structure, and they help the stabilization of this structure. And finally, there are situations in which the disulfide bond doesn't really seem to affect the structure at all. So there has to be some functional consequences behind it. So now that I introduced everything, I can tell you what we actually did, which was take the full protein data bank as the main reference of protein structures and extract all of the proteins with at least one disulfide bond. And then we applied frustration and disorder analysis to them. The results are displayed in this plot. And here you are seeing what we call the disulfide bond landscape, which are all these proteins. And you can see how the average disorder is related to the length of the protein, and the intensity of the color is telling you how rich these proteins are. So what we want to do is characterize this landscape. We can go example by example. My first situation was this case in which the small proteins are being helped to fold by the disulfide bonds. And these are these ones in the landscape. So for us to see this, we took our smallest protein, which is this one. And I think it's pretty apparent that the disulfide bonds over here are bringing together the structure despite all of these energetic local conflicts that we saw when we measured the frustration of this structure. Now, this is not just an isolated situation. We've seen this many times across this section of the landscape, and these are the patterns that are helping us understand the consequences of these disulfide bonds in these situations. We can go back to the landscape, and if you remember my second situation, it was a case when disulfide bonds were helping the folding of disordered proteins, which are basically located in this section of the landscape. Similarly to the previous case, we selected our most disordered protein, which is this one. Which is this one, and you don't see the video, but I think it's, again, pretty apparent that there was a disulfide bond bringing together the structure, although it was predicted to be disordered all along. And once again, this is happening many times across this section of the landscape. And these are the patterns that we are starting to see to explain this section. And finally, I explained that there are some cases in which the disulfide bond doesn't really seem to alter the structure at all. And for us to see these situations, what we did is create a subset of proteins that have been crystallized twice, one in the reduced state and one in the oxidized state, and that is without and with the disulfide bond. In this plot over here, you are seeing the comparison of the energetics of the contact between the two cysteines in both states. So that is without and with the bond. For you to see one more example, we selected one of the cases in which we transition from a very stable, minimally frustrated contact between these two cysteines to a very unstable, highly frustrated contact between the cysteines when the disulfide bond is present. This is the protein in question, and if we remove all of the contacts that we don't really care about, we are left with this. And I think now it's pretty clear that when the disulfide bond is not present, the contact is very energetically stable, but upon the formation of the disulfide bond, the contact distance between the two cysteines is reduced and the contact becomes highly frustrated. Now, if you see this structure is not really changing largely, so the hypothesis is that there has to be some functional consequences behind this. And if we dig deeper in the literature, we will find out that actually this protein is only able to bind to its primary partner, which is gold, when it's reduced. And the formation of the disulfide bond, far from altering the structure drastically, what it's doing is modulating this affinity of this protein towards gold and actually having some functional consequences. As a final note, I wanted to tell you that we did a conservation analysis on these three scenarios, and we found out that the cysteines that take part in these disulfide bonds are highly conserved across the species, highlighting the importance of the mechanisms that we try to characterize in this project. As a final conclusion, I just wanted to say that this is only the start of the project, so we have only started to describe this landscape, but I think it's pretty fair to say that so far we can use these frustration and disorder patterns to understand the consequences of disulfide bonds in proteins. With this, I want to thank everyone that helped me through this project, with a special mention to my two thesis directors, Professor Alfonso Valencia and Dr. Gonzalo Parra, and also to every one of you for making it to the end of the conference. Thank you.
Thank you very much. We have time for a question or two.
Thank you for the talk. So in the case of the gold binding, the gold would have the same effect as the disulfide bond in the, like, when considering the frustration of the cysteine. So after the binding of the gold, it would be like the same context as having the disulfide bond or not, like in this case.
Well, if I understood your question correctly, I don't really know how the frustration of the contact is in the presence of gold when it's bound to it, but I know that the only situation possible for this protein to bind to gold is in the reduced state when this contact is minimally frustrated.
Okay, thanks.
Okay. We will move on to the next. You have a question?
So, did you try to validate this some experimental results? I mean, there could be some proteins which
well, actually, the gold example I just explained has been validated experimentally. And some other, the examples I explained at the beginning, where we described the three possible scenarios of the disulfide bonds, are all based on experimental evidence. And what we are trying to do is to map these patterns to try and find out these situations in the landscape.
Thank you.
Thank you.
Okay.